Hello everyone, welcome back to the class. In our previous class, we discussed about DNA damage response pathway, that is ATM and ATR pathway, which plays a significant role in error correction and making DNA replication a smooth functioning process. So ATM, ATR pathway are very much distinctive in their activity and binding factors. So let's see. DNA damage response signal pathway, ATM and ATR. ATM is ataxia telangiectasia mutated. ATR means ataxia telangiectasia and RAD3 related. These are the closely related kinase, serine 309 protein kinase, which phosphorylates the particular protein. Both these pathways are activated by DNA damage. The DNA damage may be exogenous in source or endogenous in source. These are the members of phosphatidylinositol 3' kinase like kinase, that is PIKK family. In response to DNA damage, eukaryotic cells activate elaborate cellular networks collectively termed as DNA damage response. DNA damage response consists of several key components, that is DNA damage sensors, signal transducers and effectors, and at last it releases some response. So these signaling pathways lead to a DNA repair, cell cycle arrest and eventually apoptosis. ATM pathway is known as ataxia telangiectasia mutated pathway. It responds to DNA double strand breaks. ATM pathway, it is senses the double strand break, and mRNA is a sensor. mRNA binds to the DNA damage, that is double stranded break, and then it recruits ATM. ATM binds to mRNA then, and that ATM binding with the mRNA activates the downstream kinase, that is CHK2. It results in phosphorylation of kinase CHK2. Then effector, it moves to the effector P53, and the outcome will be checkpoint arrest. This is ATM pathway. ATR pathway in other context, ATR pathway responses to DNA damage, that is single stranded break, where the sensor molecule, that is RPA, replication protein A, binds to the single strand break, and it is a single strand DNA binding protein. After the sensor binds to the single stranded break, there is another one protein, that is ATR interacting protein A-trip, binds with the RPA single stranded DNA complex, and that recruits ATR to the single stranded DNA to phosphorylate, and it activates the downstream kinase by phosphorylating CHK1. Then CHK1, with the response of the CHK1 effector CDC25 will be activated. Then it is signaling, it starts the signaling transduction pathway, and the outcome will be checkpoint arrest. So these two pathways are very much important in order to repair, if possible, or cell cycle arrest. If not, at last apoptosis, that is the programmed cell death. So in today's class, we will be learning about the role of Rb protein in cell cycle regulation. So we learnt many of the regulatory proteins like what Cyclin CDK complex, mCDK. So many regulatory proteins and many regulatory enzymes, ubiquitin ligase enzymes, FCF, all these factors regulated the cell cycle and cell cycle progression. So there are two major proteins, that is Rb protein and P53. P53 will be dealt in the next class. So today we'll confine ourselves to role of Rb protein in cell cycle regulation. So in this module or in this session, you will be learning about what is Rb protein, which gene codes for Rb protein, and what is the function of Rb protein, and how the product of Rb1 gene regulates cell division, what happens if Rb gene is mutated, or what happens when Rb gene is lost in the cell cycle, and what are the significance of Rb gene in cell division. Let us know one by one. So to begin with, Rb, that is Rb protein. So we'll start from gene. So Rb stands for retinoblastoma. Retinoblastoma. So Rb is called as Rb1 commonly. So Rb1 means retinoblastoma 1. So Rb is a gene. I'll write it in a flow chart. So Rb, retinoblastoma 1, is a gene that codes for a protein Prb. Prb is what? Prb is protein, retinoblastoma protein. So this Rb, what is Rb1 here? It is a gene. Okay, this Rb1 gene is a retinoblastoma gene, which is also known as, or which is commonly known as, tumor suppressor gene. Tumor suppressor gene. By this term you may come across a simple idea, that is, it suppresses tumor growth. Just remember this, tumor, it suppresses the tumor growth inside the cell or inside the organism. The genes whose inactivation, so if this gene is active, it suppresses the tumor growth. If gene is inactive, it may contribute to development of tumors. So active, if the gene retinoblastoma is active, then it codes for protein Prb, and that suppresses the tumor growth. Okay, if the gene retinoblastoma 1, if it is inactive, so then it may lead to development of tumors. So therefore it is called as tumor suppressor gene. So what about the function of Prb? Prb is protein which is coded, or yes, it is coded by gene, that is Rb. So the protein encoded by this gene, this protein is a negative regulator of cell cycle. Negative regulator of cell cycle. Negative regulator means it stops something. It stops something. So stops some phase, we can say, phase of cycle. It stops a phase of cell cycle. It arrests the cell cycle at a particular phase. Which phase, when and where, you'll come across in just few minutes. So this was the first tumor cell. Which was the first tumor suppressor gene? So Rb1 was the first tumor suppressor gene to discover. So loss of mutation. So loss of function mutation of Rb1 gene is associated with retinoblastoma and other tumors. So loss of function mutation of Rb1 gene leads to retinoblastoma and many other tumors. In humans, RB1 gene in humans, RB1 gene is located on the chromosome 13q14.2. Remember this. In humans, where the RB1 gene is present, in humans, RB1 gene is located on chromosome 13q14b. Okay, the PRB protein encoded by RB1 gene is a nuclear protein. So the protein encoded by the RB1 gene is a nuclear protein, and its size is 110 kilodalton. Size is 110 kilodalton. Based on the sequence similarities, there are two other members of RB family. Two other members of RB family that is identified are RBL1 and RBL2. RBL1 codes for a protein P107. RBL2 codes for protein P130. All three RB family, that is RB1 also. RB1 codes for PRB. Okay, all these three RB family members contain a conserved domain. That conserved domain is known as pocket. Okay, that the conserved domain, which is referred to as pocket, that will interact with LXCXE motif, which is found in viral protein. For this reason, RB family is also called as pocket protein family. As it is having a conserved domain called as pocket, it is known as RB family is known as pocket protein family. So now, how does the product of RB1 gene regulate cell division? So far we discussed about what is RB protein, what are the types, and what function it does, and what happens if this RB gene and RB protein is absent. So what is relating to cell division? What is the factor which relates to cell division here? So the RB1 gene RB1 gene, I'll take it from top. RB1 gene codes for PRB protein. This P refers to protein here. P refers to protein. Okay. RB1 gene codes for protein PRB. So the phosphorylation of PRB protein decides the fate of cell division. It regulates the cell division. Let us know how. So there are three kinds of phosphorylation. Hypophosphorylated, hyperphosphorylation. Phosphorylation is nothing but addition of phosphate. Hypophosphorylation, little bit addition of phosphate group. Hyperphosphorylation, large amount of phosphate group is added. So here there is another one, unphosphorylation. Unphosphorylation means removal or absence of phosphate group. Here unphosphorylation and hypophosphorylation are meant to be active. Hypophosphorylation, unphosphorylation or unphosphorylated form are meant to be active. So unphosphorylation, hypophosphorylation of what? Unphosphorylation, hypophosphorylation of PRB protein. Imagine, I'll draw now. So PRB protein is there, PRB protein is there. So in first PRB there is no phosphate, so it is unphosphorylated PRB. So I have added phosphate group to the second PRB protein. So this is hypo. Okay, I have added more number of phosphate group to third PRB, that is hyper. You got my point. Unphosphorylated PRB, hypophosphorylated PRB are active. Hyperphosphorylated are inactive. Remember this logic. Okay. Active means what? Un or hypophosphorylated form. Un or hypophosphorylated form. I'll represent it like this. So this is un or hypophosphorylated form of PRB protein. This binds with, I'll write it in a different color. This binds with a transcription factor, that is E2F. It binds with the transcription factor E2F. This complex represses gene transcription. Represses means stops gene transcription of what? The gene which is required for, here comes the cell division part. This represses the gene which is gene transcription which is required for transition from G1 phase to S phase. The hypophosphorylated or unphosphorylated PRB protein binds to E2F. This complex stops the gene transcription of the which is required for what? Transition from G1 to S phase. So what does it mean? So the cell cycle is arrested at the late G1 itself. So there is no S phase. There is no continuation after G1 phase. The E2F which binds to the un or hypophosphorylated PRB, this acts as a transcription factor. E2F acts as a transcription factor and it induces many genes to transcribe. Only when it is free or only when it forms complex with others. So it is a transcription factor which induces many genes to transcribe. So the product of these many genes which is induced by E2F to transcribe, this product of these genes are required for S phase. Now that is E2F is bound with the hypophosphorylated or unphosphorylated PRB protein. So this is not inducing genes to transcribe. So there is no point of S phase here. So 5 main E2F families members have been identified, and these molecules show preferential binding to different types of RB family members. So E2F activity is controlled by PRB protein. Okay, so we know unphosphorylated PRB and hypophosphorylated PRB are active. Okay, this binds with E2F. Now we studied this. So these are active. Okay, who is phosphorylating PRB? Who is phosphorylating PRB? The cyclin CDK. Cyclin CDK complex. So the phosphorylation of PRB protein is by cyclin CDK complex. Which cyclin CDK? Cyclin CDK 4 or 6, cyclin CDK 4 or 6 and cyclin ECDK. Cyclin ECDK 2. When does this cyclin CDK 4 or 6 cyclin ECDK 2 will phosphorylate? It phosphorylates in the G1 phase. It phosphorylates in the G1 phase. So phosphorylation of PRB protein is initiated by cyclin CDK 4 or 6, cyclin ECDK 2 in G1 phase. The E2F activity is controlled by PRB protein. How? So, if there is phosphorylation of PRB, then it reduces the affinity for E2F. So, imagine PRB is alone here. Therefore, it has formed friendship between E2F. Okay. This is unphosphorylated condition. Here there is another case where PRB is having only 2 phosphate friends. Then also it is forming bond with E2F. Imagine there is more number of phosphate friends for PRB. It is engaged with all these friends. So it will not bind to E2F. E2F is now free. So there is no bond between E2F. So then what happens? E2F will form a complex with DP. DP is DNA binding protein. So this hyperphosphorylated PRB will allow E2F to heterodimerize. I'll write like heterodimer. Okay, heterodimerize with factor DP. So now as E2F has formed heterodimer with DP, there will be a function obviously. So what is that function? So E2F DP complex plays a very crucial role in the critical late G1 phase. So this induces the expression of what? E2F DP complex induces the expression of critical late G1 genes. So it induces the expression of critical late G1 genes. Why it is needed? So this late G1 genes synthesizes products which are required to prepare the cell for the entrance of S phase. So the late G1 genes are needed, whose product which is required to prepare the cell for entrance into S phase. So if there is hyperphosphorylation condition, E2F will not bind with that. Instead, E2F forms heterodimer complex with DP and allowing the cell cycle progression. We can say this is positively regulating. Okay, this is E2F is a transcription factor which induces many genes to transcribe. We discussed, no, that is genes of S phase. Now, as I said, who is phosphorylating PRB? Phosphorylation is done by cyclin CDK, cyclin CDK 4 and 6, cyclin E CDK 2. Cyclin E CDK2 is also for helping in phosphorylation. Let us now know the particular function of both of these. So cyclin CDK, cyclin D CDK, this is cyclin D CDK, okay, cyclin D CDK and cyclin E CDK2 controls the degree of phosphorylation, means hypophosphorylation or hyperphosphorylation. This types of phosphorylation is dependent on cyclin D CDK 4 and 6, cyclin E CDK2 activity. Okay. This cyclin D CDK 4 or 6 drives the phosphorylation, drives the PRB, especially hypophosphorylation at mid G1. Hypophosphorylation by cyclin D CDK 4 and 6 is done especially in early and mid G1. So it is very much needed. It is a prerequisite. It is prerequisite for restriction point. It is the prerequisite for restriction point. So what did we learn now? So we learned that cyclin D CDK 4 or 6 phosphorylates, that is hypophosphorylation, is done during early and mid G1 phase, which is very much needed, which is a prerequisite of the restriction point. So there is a significance of this restriction point. It is also known as R point. Okay, this ensures that this transition is responsive to extracellular signal. So it is a transition. Okay, and what is the function of cyclin E CDK2? Cyclin E CDK2. So cyclin E CDK associates with CDK2, or we can say cyclin E levels are increased dramatically. The cyclin E associates with CDK2. Cyclin E along with CDK2 help this complex drives in PRB phosphorylation to completion, leaving PRB protein in a hyperphosphorylated state, that is inactive. It completely phosphorylates the PRB protein. So cyclin D CDK 4/6 initiates the phosphorylation. It just hypophosphorylates in early and mid G1. But what does cyclin E do? Cyclin E associates with CDK2 and this complex drives PRB phosphorylation to completion and it leaves PRB in its hyperphosphorylated which is functionally inactive. We mentioned the R point, restriction point, right? This point represents a point at which cell commits itself to complete the rest phases. Okay, complete the remainder, we can say complete the remainder cell cycle. Okay, there are two families of CKI. So what are these CKIs? Cyclin CDK inhibitor protein. So these are CDK inhibitor proteins. So this inhibits, so the CKIs have been identified that blocks the kinase activity of DCDK 4/6 ECDK2. So we discussed the two families, that is INK family and CIP-KIP family, if you remember this family. INK family had P15, 16, 18 and 19. CIP-KIP families had P21, 27, 57. Okay, this CIP-KIP CKIs will bind with either cyclin D CDK, cyclin E CDK2 or cyclin D CDK2 CDK 4/6 complex. This INK family is believed to bind preferentially to CDK 4 or 6. So CIP-KIP family binds to both ECDK2 and DCDK 4 or 6. INK family binds preferentially to DCDK 4/6 or 6 that blocks kinase activity. So the scheme reveals that why PRB is such a critical player in the regulation of cell proliferation. If its function is lost, what happens? If the function of PRB is absent, what all consequences is resulted? So there is no longer guardian of G1 S phase transition. Imagine there is a defect in DNA. Okay, imagine there is a defect in early stage of cell at the G1 phase. So that defect will continue till the end. So loss of PRB protein functions induce cell cycle deregulation, and they may lead to they may lead to malignant phenotype. Malignant phenotype. The PRB protein can also be functionally inactivated by mutations. So by mutations or viral oncoprotein binding. So PRB function can be lost through mutation of RB gene. So that is as seen in retinoblastoma. So retinoblastoma is due to inactivation of RB gene. And there is another one, that is all osteosarcoma. That is also loss of function mutation of RB gene. And small cell lung carcinoma. Small cell lung carcinoma. All these are caused by the loss of mutation of RB gene. So now let us quickly go into the concepts. Okay, role of RB protein in cell cycle regulation, that is RB1, retinoblastoma 1. It is a tumor suppressor gene, genes whose inactivation contribute to development of the tumor. And this RB1 gene codes for protein called PRB, which is nuclear protein of 110 kilodalton, and it is a negative regulator. Loss of function may lead to retinoblastoma and many other tumors. Active hypophosphorylated form of PRB protein bind to transcription factor E2F. This represses gene transcription required for transition from G1 to S phase. E2F acts as a transcription factor and induces many genes to transcribe. Products of these genes are required for S phase of the cycle. PRB protein plus E2F makes E2F activity is suppressed. Phosphorylation of PRB reduces affinity for E2F, leaving that E2F to heterodimerize. So with DP, PRB protein is a substrate for cyclin CDK complex in order to phosphorylate. Phosphorylation of PRB protein is initiated by Cyclin D CDK 4/6 and Cyclin E CDK 2 in G1. And this PRB protein, when it makes the E2F alone, so that E2F heterodimerizes with factor DP, DNA binding protein. It forms this E2F DP complex, induces expression of critical late G1 genes. The product of G1 genes are required to prepare the cell for entrance into S phase. So this is the image depicting the phosphorylated, unphosphorylated RB and its consequences. Un or hypophosphorylated PRB protein blocks cell cycle progression. Cyclin D and E control the degree to which PRB is phosphorylated. Cyclin D CDK 4/6 phosphorylates, hypophosphorylates in early and mid G1 phase. It is a prerequisite to the restriction point R point transition in late G1 phase. Cyclin E level increases dramatically. It associates with CDK2 and drives PRB hyperphosphorylation and makes it inactive. R point is a restriction point. It represents a point at which the cell commits itself to complete the remainder of cell cycle, or alternatively to remain in G1 and possibly retreat from the active cell cycle into G0 quiescent state. So it acts as a main point. So there are two other RB family that has been recognized, and it is therefore known as pocket protein family, as there is a conserved domain which is referred to as pocket in RB1. So there are RB1, RBL1, RBL2, and it has a conserved domain pocket which interacts with LXCXE motif. And that's all. So in next class, we'll be learning about role of p53 protein in cell cycle regulation. Thank you.